Cytoskeleton. Another principal component of cytoskeleton, apart from microtubules and microfilament, is the intermediate filaments. And these set of cytoskeleton are more responsible for integrity, structural support, and shape rather than movement, because these intermediate filaments are not really associated with molecular motors, unlike microtubules and microfilaments. So let us look at the learning outcomes of this session. So unlike microtubules and microfilaments, the intermediate filaments may not be part of every cell type. These are of a size which is intermediate between microtubules and microfilaments, and hence the name intermediate filaments. Microtubules are 25 nanometers, microfilaments are 7 to 8 nanometers, and intermediate filaments are around 11 to 12 nanometers in size, and therefore they fall between microtubules and microfilaments. So intermediate filaments are of several types, in fact. The reason for them being several types is that they are made up of more than one different protein. Unlike, for example, microtubules are primarily made up of tubulins, microfilaments are made up of actins, but intermediate filaments, different types, are made up of different proteins. And they are associated with specific cell types as well. It is not necessary that you will find all types of intermediate filaments in all cells. Unlike microtubules and microfilaments, intermediate filaments do not have polarized structures, that is, they do not have two distinct ends, and they do not have binding sites for nucleotides, so they are not associated with either GTP or with ATP, and nor are they associated with molecular motors. So in these many ways, they are way different from microtubules and microfilaments per se. Several of the intermediate filaments have been well studied, although there is much more to know about intermediate filaments, and in fact, new types of intermediate filaments are being discovered over a period of time. The nuclear lamina is one of the intermediate filaments that has been well studied, as it is also associated with the cell cycle, with cell division per se, and so it has been studied. These are able to structurally and functionally help the nucleus, especially during the cell cycle. So let us look at the primary structure of the intermediate filament. So whatever type of proteins it is, the basic structure or the way the structure of the intermediate filament develops is more or less the same. So generally one finds that the intermediate filament is comprising of monomers, and these monomers are very typically having a central helical region. So as you can see over here, this structure, which is a monomer, is polarized. Why do we call it polarized? Well, because you have a COH end, which is different from the NH2 end. So therefore, as a monomer, they are polarized structures, and also the central helical region is conserved. It is found to be highly conserved. When you have a dimer being formed now, the dimer forms again a coiled dimer, just like the actin filament does. Uh, and in fact, when the dimer comes and binds, again the dimer is bound in the same orientation where NH2 and NH2 are the same face, and the COH and the COH make the same face. So therefore, as a dimer as well, the structure is polarized, with one end being negative because of the presence of carboxyl group, and the other end being positive because of the presence of the NH2 group. However, the equation changes when you have a tetramer being formed. So there are two important aspects of this. One is that the second dimer, when it comes to bind, the orientation of the second dimer is exactly opposite to the orientation of the first. So therefore, the NH2 and the COH of the second dimer come together in a tetramer, and therefore the plus of the NH2 and the minus of the COH negate each other. So there is no net charge on the on this end, nor is there a net charge on this end. So therefore, eventually you would have both NH2 and COH present on either sides of the tetramer. So effectively what we understand is that as a tetramer, the structure is no longer polarized. Neither do you have a net charge on either side, nor do you have different functional groups present on the two ends. And another important aspect is that the binding of the dimers to each other is in a staggered form. So you will not find that the COH ends are completely aligning with NH2. In fact, you will find that it is present at a certain distance. So you have the NH2 of one of the dimer being an overhang, and in the other also you will find the NH2 being a slight overhang. So that is how you will find that they are staggered ends. They are not blunt ends. They are completely staggered ends. So this is important because this kind of becomes a hook for another tetramer to come and bind that way. So looking forward to how now the filament is structured, because we've just reached till the tetramer per se. So what is observed is that you can have to this, this acts as a hook. So the other, so another component can just come and bind such a way that one staggered end binds to the other staggered end. So you will have another staggered end binding over here. So that way you can have several sets of the dimers coming and binding, but it does not bind as dimers. Rather, it what it happens is that 8 tetramers come together. So you see, this is one tetramer. Likewise, you have 8 tetramers coming together. They bind to each other laterally, and they kind of, like you roll a carpet. That is how they are rolled, okay, to form a structure like this, a cylindrical structure like this, where the staggered end of this is present over here and the staggered end of this is present over here. Now, such tetramers which have formed now a complex, okay, you would have several tetramers coming and binding one end to the other end like that, okay. And so now what you have is generally a big intermediate filament formed, with each 8 tetramers being the base, or being the monomer of the entire filament. So effectively what you will understand over here is that this intermediate filament has a lot of mechanical stability, and instantly you would think of the fact that it is easy to break one twig, but when you have several twigs put together, it becomes difficult to break the twig. So that is why this intermediate filament has strength in it, okay, and so it can resist forces, it can give structural support. So that is the main integrity of the cell is definitely being maintained by the intermediate filaments per se, and this is how most of the intermediate filaments are present as. But as you can very clearly see, you do not have a plus end or a minus end, or sorry, you don't have a polarized structure basically. Can you have the intermediate filaments undergoing dynamic instability? So the answer to that is yes, they have found that intermediate filaments can also assemble and disassemble, but not to the extent that microtubules and the microfilaments are carrying out dynamic instability. But yes, you can have assembly and disassembly. Now we go to different types of intermediate filaments. So type 1 is the acidic keratin and type 2 is neutral or basic keratin. So you will see that both are basically keratin. The only difference being that type 1 has keratin that is made up of a higher percentage of acidic amino acids. So its overall nature is acidic, while type 2 has a higher percentage of basic amino acids or non-polar amino acids, neutral amino acids rather, and therefore they conform to what is called as a type 2 intermediate filament. Interestingly, you can have various different keratin coming together to form heteropolymers, and in fact that is the reason why keratin has a lot of biodiversity in terms of its structure, in terms of its presence in different cell types. As you can see in this picture very clearly, the blue fluorescent tags are used to tag the keratin, and you can see how in a cell you have the keratin right from the nucleus to the cell membrane, kind of forming a structural network that is giving shape to the cell and also support to the cell. Plus, what you would observe is keratin of one cell is able to interact with the keratin of the other cell through cell-cell interaction, especially what is called as tight junctions. So you also have therefore cells, especially epithelial cells, bound to each other to form a sheet, and the cells are stitched to each other, and inside what you have inside the cells connecting two cells is basically the keratin itself. So an epithelial cell can produce several keratins that can form a mesh-like network, and keratin of one cell are indirectly connected to the keratin of the neighboring cells, thereby making what is called as cell-cell interaction. Also, bundles of keratin form the top layer of the skin. So you can call the skin as having a keratinized layer, and that imparts toughness to the skin. Defective keratin has been associated with several diseases. Especially, you will find that when you have improper keratin being formed, then there are blisters on the skin, very visible blisters that are present on the skin. Now we go to the second type. Second type is what is called as the neurofilaments. Again, neurofilaments, there are three types: NFL, NFM, and NFH. And these three actually can combine with each other to give rise to heteropolymers. So, effectively, as the word suggests, neurofilaments have been found to be present in neurons, especially in the axon region. So, this that you see over here is axon, and neurofilaments are able to interact with the other two cytoskeleton also. So, you can see that it can interact with the microtubules. So, the light blue are what are the neurofilaments, and they can interact with the microtubules. They can also interact with the actin, and they can also help the microtubules carrying out transport. So whether the transport across the axon is fast or it is slow is actually dependent on how the neurofilaments associate with the microtubules and the actin filaments per se. So as you can see over here, you can have the neurofilament have certain proteins that can directly bind to microtubules, and microtubules therefore can be associated with the neurofilament. So you basically have an interconnection that is there between neurofilaments, actin filaments, and the microtubules in neurons, and they work in tandem to carry out the functions in the neurons. Like for example, here it is very clear that the neurofilaments are able to interact with the molecular motors that are working on the microtubules, and based on that you have the neurofilaments actually push towards a certain direction, and that kind of helps in regulating direction, in regulating transport of molecules across. And not just that, it is also giving a certain structure to the axon itself, because it has been noted that if the neurofilaments are defective, then the axon can be damaged, or the shape of the axon is not proper, and because that is the case, you would have transport across the axon being compromised, and therefore the neuronal function gets compromised per se. So these are certain things that are associated with neurofilaments. Then you go to the next one, which is Vimentins. For example, desmins, and these desmins are found in muscle cells, but there are other types of Vimentins also. But here we are taking the example of desmins, which are found in muscle cells. So as you can see over here, the desmin is actually acting as a scaffold, okay, around the Z disc. So we know that the sarcomere has a Z disc. So you see cap Z over here. A group of cap Z forms what is called as a Z disc. So the desmin forms a scaffold for the Z disc. You can also see that the desmin is associating with microtubules. It is associating with the muscle membrane. It is associating with the microtubules. It is associating with the actin filaments. So therefore, it is making an intermolecular network which helps the muscle functioning. So it has in fact been observed that if there are defective desmins, then it has been associated with diseases like muscular dystrophy and cardiomyopathy. The fifth type, which has been studied to a great extent, is lamins. There are actually three types of lamins that are present, lamin A, B, and C. And as you can see, this is the nuclear membrane. And just below the nuclear membrane, you can see the three lamins are associated as a mesh. Again, they form what is called as a scaffold. And in fact, the word lamins comes from the fact that it is laminating the nuclear membrane from inside. So you see, you have your mark sheets now, because you want to preserve the mark sheets for a long period of time, you would go and get it laminated so that it is protected or so it is supported or so that the paper gets a certain, it doesn't form creases, etc. So that way you can understand in layman's terms that the lamins are actually laminating the nuclear membrane from inside and therefore providing a support for the nuclear membrane. So the nuclear membrane does not really collapse. Secondly, the lamins is enabling give the nucleus its spherical shape. So because it forms a scaffold, it is also giving the membrane a support to be in, and the way the lamins are arranged decides how the shape of the nucleus is. Moreover, the lamins are associated with several other different proteins which are called as lamin associated proteins. So you can already see so many different proteins that are associated with the lamins. And interestingly, these lamin associated proteins allow the nuclear membrane indirectly to be in connection with the genetic material. Here what you see is the heterochromatin, and it is also in connection with the cytoskeleton outside the cell. So there is a connect that is there between the cytoplasm and the nucleoplasm, and these lamin associated proteins enable signal to be transferred from outside to inside or from inside to outside. So there is inside out signaling and outside in signaling thanks to the interactions that the lamin associated proteins make with components in the cytoplasm and components within the nucleus. So effectively, the lamin is also a scaffold for proteins that help in interacting or the proteins that are present in the nuclear membrane, and in one way or the other they help in signal transmission, signal transduction, and also interaction of the nucleus with the surrounding environment. So that is very, very important, and the lamins therefore play a very important role. Now, we all know that when cell division has to occur, then the nuclear envelope kind of disintegrates during pro-metaphase. Pro-metaphase. So you have the nuclear envelope completely getting disintegrated into small vesicles, and it is present suspended in the cytoplasm. So these lamins, which are holding the nuclear membrane, actually at that time the lamins get disintegrated or they disassemble from each other, and thereby along with them getting disassembled, they carry with them the nuclear membranes and exist in the cytoplasm as small vesicles. So therefore the lamins, are regulated to form the nuclear envelope as well as to disassemble the nuclear envelope when required by, of course, regulatory molecules. Now, sixth type of intermediate filament is also being considered, which is the nestin. And nestin is a connecting protein that is present between microtubules present in the flagella and the cilia. So, likewise, there are several other types also being studied, but one doesn't ascertain that they are intermediate filaments as of these. The type 5 intermediate filaments have been studied to a great extent. So with this we make the conclusions. The intermediate filaments are made up of different proteins, with its major role being in imparting structural support and integrity. Depending on the type of proteins constituting the intermediate filaments, they have been classified into several types. Unlike microfilaments and microtubules, the intermediate filaments generally do not have polarized structures and are not associated with molecular motors. The intermediate filaments, in comparison to microfilaments and microtubules, are more stable structures. So we are not saying that it does not exhibit dynamic instability, but in comparison, it is definitely much more stable than microfilaments and microtubules. So the intermediate filaments mediate several functions in different cell types. They act as scaffolds and support systems apart from being involved in cell-cell interactions and cell signaling. Thank you.